today I'll be talking about a statistical approach for removing joint and individual unwanted variation from single-cell multi-omics data. So I'll start by talking about the multi-modal analysis using transcriptomics and proteomics data. Then I'll address the importance of removing unwanted variation from the data and what are the current methods for this task. Then I'll introduce our approach and show some benchmarking results, and finally the discussion. So there are many platforms for profiling single-cell transcriptomics and proteomics. And one of the most commonly used assay method is droplet-based. So basically, the experiment starts by binding antibodies to the cells, and the cells then travel through the pipe together with some microbeads, and they are encapsulated with oil drops. And ideally, there's only one cell and one bead in a droplet, and the cell membrane would get destroyed during the cell lysis. The cellular materials such as RNA and protein molecules are captured by the reverse transcription oligos on the surface of the microbead. And this is how we can simultaneously quantify cell surface proteins and mRNA within a single cell. So there are some sequencing-based assays like CITE-seq and RIP-seq, and those have this droplet-based protocol, and the analysis of the multimodal data can sometimes reveal more biological insight. So an example is that we get a more refined cell types. In this example, that is CD4 T cells, when we perform the joint analysis using the features from different modalities. So, and here, I would like to emphasize the importance of removing unwanted variation. So the sources of unwanted variation can range from domain-specific library size and other technical variation. And if that's ignored, it can lead to misleading conclusions from the downstream analysis. So this is an example using the uncorrected data. And we see two CD4 T cells, CD8 T cells clusters on the UMAP coordinate. And we might go, oh, we found subtypes of CD8 T cells. However, if we color the cells by the batch they came from, then we know that they are from different batches. And it's really hard to say whether this is a true biology or not. And we will come back to this example later after correcting the data. So there are some existing methods for removal of unwanted variation. And usually those methods start by data normalization. So, and the normalization is done batch by batch for each domain separately. So those are some existing methods. However, those methods sometimes cannot remove batch effect from the data. So we need an extra step, which is step 2, to deal with the batch by integrating batches within each domain and using methods such as Seurat anchor strategy or harmony. And for further modality integration can be done by Seurat weighted nearest neighbor method. So, and the issue of this procedure is that we don't get the corrected data at feature level after the step 2. Plus the batch information, we need the information in order to deal with batch. So our objective here is that we want to obtain the corrected data at feature level without using the batch information. And this will enable the analysis, which is impossible to be conducted with the embedding level data. Okay, so our approach, everything stems from IUV3MB approach, IUV3MB model. So for the feature F and cell C, we assume the UMI count is coming from a negative binomial distribution with mean parameter mu, and the dispersion feature-specific dispersion parameter psi. And for each feature f, we model the mean parameter. And this is the vector for n cells. So this is what our generalized linear model look like. And this model consists of several parts. So we have one part here, the M matrix we need to specify for the model, and M matrix contain the biological information of the data set. And here we have M subpopulations in our data set. And for the other parameters, such as the intercept and the unwanted factors matrix W, we don't need to know the parameters here. And the IUV3MB algorithm would estimate that for us. So, and beta and alpha, they are the unknown effect size of biology beta, and the unwanted factor effect size of unwanted factor alpha. So the advantage of using this generalized linear model is that if we have proper negative controls, so negative controls, they are actually the features that capture unwanted variation in the data. And we will use that to estimate the W matrix in the model. And also we only need a subset of replicate, at least 1%, which means we don't need every single cell in the data set to be annotated. We just need a portion of cells, the information of the biological information of the cells in order to get IUV3MB running. And also we have to specify the number of unwanted factors that we want the IUV3MB algorithm to estimate. And this model, another advantage of using it is that it's better for the case when biology and unwanted variation are associated, because here we account for biology in a model. So we estimate the parameters in the generalized linear model through the modified iteratively reweighted least square. And the concept is that this algorithm contains a double loop. So firstly, we set up initial parameters for the initial estimates for the parameters. And given the current estimate of the dispersion parameter, the algorithm would update the parameters of the log linear model. So the alpha, W, and the intercept and the beta will be updated in the inner loop. And after that convergence, the dispersion parameter is then updated in the outer loop. So in this algorithm, we work on the working linear model. And this is actually the first order Taylor approximation of the generalized linear model that we mentioned above here. And we update the parameters one at a time using the closed form that we derived from the likelihood function. And then in order to estimate W matrix, we make use of negative controls. And this is why we call the iteratively reweighted least square a modified version. And for the negative controls, we assume its biological effect to be 0. So we don't have this term in the working linear model for negative controls. So all the details about the closed form for updating the parameters can be seen in the IUV3MB paper. So for the multi-omics data, we modify the current IUV3MB structure that I mentioned in the previous slide to accommodate the shared and distinct unwanted variation. But before I introduce the structure of the extended IUV3MB, here the question is, why don't we just apply IUV3MB directly by stacking all the features as input? And the advantage of this is that we just need to apply the current IUV3MB model. We don't have to develop a new method. But this advantage of this is that when it comes to estimating unwanted factors W, it's impossible to separate the unwanted factors from different domains. because they all come together. And there's potentially a problem is that the computation during the estimation process, like for example, some protein features can be more variable than RNA features. So here is the structure of the extended IUV3MB. So basically, we have the same broad structure as IUV3MB, but we did some modification for the unwanted components, W and alpha. So the W consists of 3 parts, which are 2 distinct unwanted vector matrices for the 2 domains, RNA and ADT proteins. And W3 represents the shared unwanted vectors. And the structure of the corresponding effect size alpha depends on the type of the feature. So for an RNA feature, it won't have the unwanted effect from the protein domain. So we have a 0 in the middle. And the same logic for the protein features. And there are some methods developed for the quantification of shared and distinct information, but they were designed for the composition of biology, like Jive and tilted CCA. So if we apply them to our context, using tilted CCA can help us achieve the orthogonality between the 2 distinct unwanted vectors. On the other hand, Jive can help us achieve the orthogonality between the distinct and the shared unwanted vectors. And this is more aligned with our objectives. However, the estimation of Jive involves permutation, and that is computationally expensive. So to achieve our goal, we insert 2 constraints into the likelihood function, adapting the method of Lagrange multipliers for estimating our shared unwanted vectors. So here I'm going to give the geometric interpretation for behind the constraints. So our goal is to make the distinct unwanted vectors to be independent or orthogonal to the shared unwanted vectors. So that means in the below matrix multiplication, the element in the A matrix should be 0 everywhere. And that means when we do the matrix multiplication, the columns in the 2 matrices, we can see that in the vector space, they are actually perpendicular, and the dot product will be 0. And this is why we will have zeros everywhere in the A matrix. So, and the same logic can be applied for looking at for achieving the orthogonality between W2 and other modality and our shared unwanted vectors. Okay, so with a bit of derivation, we obtained the closed form for inferring the shared unwanted vectors with the 2 constraints that I mentioned in the previous slide. And for the other parameters, it's relatively straightforward to get a closed form for inferring the other unwanted components, W1, 2, and alpha 1, 2, 3. And for the rest of the parameters, like the intercept, the biological effect, and the dispersion parameter, we just retain the current method in current IUV3MB method. So now we are all set. And next session, I will show results from benchmarking by using these 2 data sets. So we have one Triana data set by the samples are from human bone marrow. And for Gayoso data sets are the samples from mouse spleen and lymph nodes. And for the extended IUV3MB, we set the 3K parameters to 1. And for the concatenated IUV3MB, we set up K equal to 3 for a fair comparison between those 2 methods. So for the assessment of the unwanted and biological variation in the corrected data, we calculated Lise scores. So the Lise score, basically, it indicates the number of different categories represented in the local neighborhood of each cell. And the categories can be either batch or biology. So when the Lise score is equal to 1, that means all the cells, the neighborhood cells, that belongs to the same group. So that is the smallest diversity. And whereas when the Lise score is larger, that means we have higher diversity in the neighbor area. And we also have some visualization using UMAP embeddings. And I'll show some D analysis results for comparison. So let's start from Gayoso dataset. This is the dataset when extended IUV3MB outperform others. So from the Lise score, we can see that the y-axis here represent the batch Lise, and the x-axis is the biological Lise. So ideally, a good method should give us higher batch Lise score, because that means we have a better mixture of batches. And for the biological Lise, we would expect to have lower Lise score if the method doesn't ruin the biology. So in this case, we can see that both IUV3MB method, the concatenated one and the extended one, we return a better mixture of batches, and we retain the biology in the data. While other methods, the triangle in the hollow triangles means other alternative methods. And we can see that many of them, they remove batch quite well, but at the same time, they remove the biological signal from the data. And this gray dot is a reference point from the raw data. And also I did the comparison by calculating Lise score for the Seurat integrative result. So this is how Seurat deal with batch. And it turned out to be too strong that remove both batch and biology. So here I go back to the example that I showed at the start of my talk. By looking at the raw count, we saw that the CD8 T cells, we have 2 subclusters, and we are unsure whether they are the true biology or not. And by looking at the IUV3MB corrected data, we saw that those 2 classes that merge together, the light green ones. And so this suggests that the subclasses that we found here is likely to be unreal. And also we checked the performance of other alternative methods, and it turned out to be not so good, because we can see that the CD4 T cells and CD8 T cells, they are clumped together. We can't really see the difference between those 2 different T cells from other methods. And it's even worse when we check the performance of Seurat anchor strategy to remove the batch effect. It removes everything. And another example is by looking at Triana dataset. And this is where extended IUV3MB is more conservative. So again, the Lise score, we see that this is where we can see the difference between the concatenated IUV3MB and the extended IUV3MB. So extended IUV3MB is able to retain more biology compared to the concatenated one. And from the batch aspect, we see that the IUV3MB method is not as aggressive as other methods, because some methods you can see they remove batch quite well, but at the same time they remove biology as well. So with this example, I would like to show some DE results by looking at the DE markers for differentiating CD4 T cells and CD8 T cells. So here I'm calculating the proportion of biological DE markers found from different normalization methods. And it turned out to be the IUV3, extended IUV3MB was able to have slightly higher proportion of biological DE. And on the other hand, I also check the batch DE by focusing on CD8 T cells. And we can see that the proportion of batch DE from extended IUV3MB is lower than other alternatives. So that suggests that the biological DE that we found, the proportion higher, they are likely to be real. So the current work in progress, regarding the methodology, the current constraints defined in the likelihood function have an issue of getting undefined Lagrange multipliers, and that was solved by an ad hoc method. So now we are actually developing a new strategy. So that is, we want to apply a softer constraint by taking the insight from canonical correlation analysis. And the idea is to minimize the correlation between the distinct unwanted factors and the shared unwanted factors. And also we plan to add harmony for the comparison. And this is the end of my talk, and I really appreciate my supervisor's guidance and providing helpful suggestions, and also lab members' comment, questions, and discussions are valuable as well. And also I thank Liao for sharing the resources of CITE-seq dataset. And finally, the findings from the University of Melbourne and Taiwanese Ministry of Education. Thank you, and I'm happy to take any questions.